Hello everyone, my name is Andrea Lee, the Bazuka Prophet, and today we are going to be working on a very interesting pathway which is called the Urea cycle. Now quickly, a quick rush on it. Now the Urea cycle is the first of the metabolic cycle that was first discovered in the year 1932 by two scientists, and their names are Scribe and Conencelit. Don't also forget, in the year 1937, Scribe and William Johnson also elucidated, or they were the first to describe the TCA cycle, which we also call what now? Which we also call the Krebs cycle. Please do not forget. Now we move straight away quickly that a urea cycle is a diamine, a diamine of what now? Of carbonic acid. Get it now. Unlike ammonia that is toxic and alkaline in nature, the urea is not toxic and also it is neutral. Now we continue. Now it will also interest you to know that the urea occur primarily in the liver, exclusively in the liver, although some few tests will tell you in a lesser extent it occurs in the kidney. But in my context, the urea cycle occur primarily in where? In the liver. So we continue. Now in general, it will also interest you to also know that the overall cycle of the urea, the overall reaction of the urea involves what? Involves 3 ATP. That's 3 ATP. That's 2 from here and also and 1 from the step 3 with bicarbonates and ammonia. Please take note, this ammonia is coming from the breakdown of glutamate plus aspartate to yield what we call fumarate and urea, and also the liberation of what now? Of 2 ADP plus PI and AMP, which is called the adenosine monophosphate. Now take note, in a quick breakdown of the urea, take note that these two nitrogen that you are seeing here are coming from two important substances, and they are: the first nitrogen is coming from ammonia, while the second nitrogen is coming from this aspartate. Please take note. While this carbon is coming from bicarbonate, call it bicarbonate or CO2. Now, if you look at this pathway thoroughly, you will understand that there are 5 steps that are involved in this cycle, and they are this step 1, which is A, which is step 1, which is step 2, step 3, 4, and 5, which I label them A, B, C, D, and E. Now we move straight away that in this pathway that you are seeing here, you will understand some basic facts about this pathway. And the basic facts about the pathway is that, if you observe, this two pathway occurs in 2 parts of a cell, and it occurs in the mitochondria and again too in the cytosol. In the mitochondria, only 2 steps are involved, and the 2 steps that are involved in mitochondria are step A and step B, while in the cytosol 3 steps are involved, and the steps that are involved are step C, step D, and step E. You get me now? Take note that ornithine and citrulline, for them to be able to be transported away from the mitochondria, from mitochondria, they will have to require a special transport system, which in this aspect we'll do our best to talk about them. Now, the steps, first step involve 2-ATP, which is adenosine triphosphate with a bicarbonate with ammonia, in which the enzyme involved to catalyze this first reaction is called the carbamyl phosphate synthetase, to produce what we call a carbamyl phosphate with the release of 2-ADP, which is called adenosine diphosphate, and inorganic phosphate. Is that clear? Good. Now, in the second step, which is B, please take note that the step 1 is an irreversible step. And because it is an irreversible step, it is the rate limiting step. Take note, it is the rate limiting step and also the regulatory one step. Now, when carbon phosphate is formed, it then reacts with ornithine, so that in the presence of the enzyme called ornithine transcarbamylase, citrulline is formed. Citrulline is formed. Very simple. This is the structure for carbamyl phosphate, in which when this carbamyl phosphate reacts with ornithine, this is the structure of ornithine. It's a very simple logic. You are already aware that an inorganic phosphate has already been removed, leaving you with this structure left. This structure will now combine with this ornithine, which is what you are seeing here. You see it now. You see the structure. Very easy. Very, very easy. Now, when citrulline is formed, remember I told you that it will require a special transport system to take it to the cytosol. Now, the citrulline, in the presence of another ATP, altogether a 3-ATP is used in this urea cycle pathway. So that an ATP will now release AMP, which is called adenosine monophosphate, plus a pyrophosphate. What will be formed? Adeninosuccinate is formed, which is being catalyzed by aspartate. Do not forget, as I've told you, that the second nitrogen atom is produced from aspartate in urea, and this is a structure. Urea combines with citrulline to give what we call adeninosuccinate. This is the structure of adeninosuccinate. Very simple. This structure here, which is the aspartate combined with adenine. Very simple. This is what you get. Now, adeninosuccinate, we then remove a compound or a substrate which we call the fumarate. Remember that a fumarate is a very important substrate or a precursor. Remember that fumarate can undergo a reaction being catalyzed by fumarase to give us what now? Malate. From malate to oxaloacetate. From oxaloacetate, another pathway is regenerated, which we call the gluconeogenesis. Are you with me now? So we continue. Now, when adenine is formed, take note, in the presence of water, adenine in the presence of water will give us what we call urea, which is being produced, and ornithine. Ornithine too, for it to enter this mitochondria, a special transport system is utilized, which is what you have here. Again, the cycle continues. The cycle continues. Now, in a nutshell, do not forget that this pathway is of great necessity to us, being that the excess nitrogen in our body system, if that are toxic, if they are not being removed, they can cause serious problem or a distortion or an imbalance in the human or in living organism, and as such, they should be removed or they must be removed. Remember that urea in the presence of water is what produces urine, which is being excreted via the kidney. Do not forget some basic point I told you earlier, that the urea cycle occurs exclusively in the liver. Don't forget. And that the step 1 is an irreversible step and also the rate limiting step. As such, it's a regulatory step. Don't also forget that the urea cycle, this first step, remember that this enzyme, carbonyl phosphate synthetase, is what is involved in where now? In the activation and condensation of these reactants to give us what we call a carbonyl phosphate. And the enzymes that are involved, in a nutshell, A, which is the first step, is carbonyl phosphate synthetase. The second step, ornithine transcarbamylase. In the step 3, which we call step 3 is called adeninosuccinate synthetase. And in step 4 is adeninosuccinase. And in step 5, which is the last one, is the arginase. For now, I wish you all the best, and please do well to subscribe, share to your loved ones who are into this realm of biochemistry. God bless you. Thank you so much.